Okay, thank you so much again, Angie and Brent, and to Richard for being here and for sharing your stories with us and allowing us to learn from you. We so value your time and that you're here with all of us. We are going to move right along into our Meet the 2024 John Quale Travel Fellows session. I think you're all here sitting up front, but if any of you are not already, can you please move up to one of the front tables so that you can jump up on stage easily once you're introduced. I'm very pleased to announce that Beacon again was able to award 8 travel fellowships to support early career investigators to attend the think tank meeting and be in this room with all of you and collaborate and learn and launch their careers in bladder cancer, we hope. Please introduce yourselves to them at some point during the meeting. We will have a poster session and happy hour tomorrow towards the end of the day so that you can do that and introduce yourselves. And I'm going to soon hand it over to Dr. Max Cates to introduce the award and the fellows, but first I want to thank him for serving as our Scientific Review Group Chair this year for the 2024 cycle. This is such an important award to Beacon, and Max has done a wonderful job in your inaugural year as chair, so we're very grateful to you. And you were also a fellow yourself in 2015, I think. So you've now come full circle, and if I can ask you to join me on stage to introduce the fellows, and then you can come up as Dr. Cates introduces you.
So this is going to be a really fun session because we have 8 fantastic fellows to talk to you about their work, who they are, and then we'll have an opportunity to meet them during the happy hour. But first, I really want to thank the work that Rebecca and Anne and Diane have put in to helping choose these fellows to be on the committees, but also to the reviewers who looked through these applications, and really to the reviewers in all of the programs. I may be jumping the gun from something we're going to do tomorrow, but if you've been a reviewer this cycle, this year, on any of the Beacon programs, can you just please stand up? So this is a form of service that kind of goes unsung, but I think I speak for everyone when I say it's very much appreciated. I also want to thank Petros Grivas, who I'm taking the baton from. I was his intern last year, learning this process, and I may be more succinct, but other than that, I will not be as good. So we're going to jump right in. These fellows, as was said, 8 fellows were chosen from 33 applications, and they really represent the swath of bladder cancer research within our community. The first is Dr. Joshua Linscott, who's a urologic oncology fellow at Moffitt. His project title is Tumor Fraction and Copy Number Burden from Circulating Tumor DNA Predicts Minimum Residual Disease Prior to Repeat Transurethral Resection in High-Risk NMIBC. His mentor is Dr. Roger Lee. Joshua Linscott is, as I said, a urologic oncology fellow with Dr. Lee as his mentor, and his goal is to demonstrate the utility of circulating tumor DNA in non-muscle invasive bladder cancer, and they look forward to expanding their research into muscle invasive bladder cancer, as well as several other disease spaces. In his personal statement, he said, "Receiving the John Quale Travel Fellowship Award is an honor, and I am fortunate to be a part of the legacy of recipients, many of whom have made impactful contributions in bladder cancer research. I hope to continue this tradition and look forward to the opportunity to collaborate and brainstorm with many field leaders in order to improve care for our patients."
Well, thank you. Definitely an honor to be here and be selected. I'm Josh Linscott. I am originally from Portland, Maine. I'm now at Moffitt Cancer Center. It was a little bit of a wayward path for me to get there. I was a graduate student at Weill Cornell Medical College, actually, studying epigenetics there and cancer research, and became sort of a patient through my dad becoming a patient. So while I was in graduate school, my dad had non-alcoholic fatty liver disease and ended up needing a liver transplant. And so it was sort of in the middle of me going through graduate school, and I was trying to figure out, what does my family do with this? Do I leave? Do I help him? How do we get through this? We ended up going to the Leahy Clinic where they do live donation, and so found out that you can have live liver donors. And I was fortunate enough to be able to donate to him. And so I donated part of my liver to my dad while I was a graduate student, and literally woke up with teams rounding, was like, this is what I want to do. I want to go into medicine. And so I finished graduate school sort of because of the encouragement from them, and then jumped right into medicine, wanting to find a way to really have an impact on patients. I didn't think I was going to be a surgeon. I thought, those guys work too much, I won't do that. Turns out everyone works a lot, so I loved being in the OR, and I loved the team environment. And I wanted to find some place where I fit in, and urology just felt like a really great fit. And I was preparing for a presentation as a visiting intern, acting intern, whatever you want to call it, and sort of saw where the field was in terms of research and how so much of the epigenetics and the genomics research that I'd been doing in the past was coming to the front. And suddenly I was like, oh man, this could be great. I knew I wanted to do the cancer side of things, so I was already thinking about fellowship. So I started looking at places where I could do a fellowship and sort of bring together these two worlds. And I think probably from within the first couple of minutes of talking with Dr. Lee, I was like, this is it. I was like, this guy's doing what I want to do. This is going to be great. I hope it works out. And it worked out. And so a little bit about our research. We're looking at the fact that your cells turn over all the time, right? Either because of cancer, because of normal cellular processes and cell death. And when they do that, they release some of their DNA. These little bits, these small pieces. And those bits of DNA are going to float around in whatever they're in. When they're in their bloodstream, they stay in the blood. In the bladder, they stay in the urine. And so they don't last long, a couple hours on average. And so it's a potentially great diagnostic tool. So I would say most of my research right now is focused on sort of the diagnostic and therapeutic applications. We're using cell-free DNA to sort of see if there are better ways to pick up patients from their urine, maybe actually predict what they would respond to. And so tomorrow night I'll be sort of going over the poster, which is the first project here looking at urine and non-muscle invasive bladder cancer, detecting disease by looking at actually the tumor fraction in that cell-free DNA or the copy number variation, which if you are actually interested in what that means, we can talk about it more. I think that's all I'll say.
Next, we have Dr. Andre Kidd, who's a medical oncology clinical fellow at the NCI, the National Cancer Institute. His project title is Preclinical Models, Translational Studies, and a Clinical Trial on Antibody-Drug Conjugate Regimens in Rare Bladder Tumors. His mentor is Dr. Andrea Apollo. Dr. Kidd matriculated at the Johns Hopkins University School of Medicine's MD PhD program. He then completed his internal medicine residency at Florida Atlantic University, where he served as one of the chief residents. He joined the NIH as a clinical research fellow in 2020 with Dr. Apollo prior to entering the NCI's Hematology Oncology Fellowship Program. He is currently engaged in a series of laboratory, preclinical, and translational projects, as well as clinical trials in bladder cancer under the ongoing mentorship of Dr. Apollo at the NCI. In his personal statement, he states, "The Beacon think tank provides a unique platform to engage with patients, patient advocates, and fellow clinicians and researchers that shares the mission to improve the care of people with bladder cancer. This award allows me an unparalleled opportunity to network and build new collaborations with this community that is so critical to building my career in bladder cancer research and patient care while connecting with bladder cancer patients, caretakers, and advocates."
Thank you, Dr. Cates, for that introduction. Again, I'm Dr. Andre Kidd, a third-year Hem-Onc Fellow at the National Cancer Institute, part of the NIH in Bethesda, Maryland. Again, a thank you to the BCAN Think Tank Organizing Committee, the Award Review Committee, and I'd also like to thank the BCAN staff that actually helped with the logistics that allowed me to be here. It was a little bit nuanced given my role within the federal government, so very appreciative of that. And again, taking a step back and thinking of being here under a travel support, think about the journeys that we're already hearing about that I've seen before, and why that's very apt as we're thinking about my role and how I got to this point, looking ahead to the future, and how I'm engaging with everyone that I hope to meet and learn from during the time that we have here together. Pretty early on, I knew that I wanted to do something that was able to combine my love of research and science, but something that really was going to move forward and help with patient care and improve patients' lives. Pretty early on, I realized that cancer was one of those fields, and knowing already decades ago there was lots that was going on, still realized how devastating that was. Still not specific to bladder cancer at that point, but it was a thread that led me through earlier projects through undergrad, being an MD PhD and studying cancer genetics and epigenetics, later projects, and ultimately the desire that I knew that I wanted to go into a hematology oncology program, doing some combination of aiming for both research and clinical care. In the setting of returning to the NIH around 2020, in part coming back is the undergraduate scholarship program in which we returned to the NIH after earlier times being there. I connected with Dr. Apollo, who actually was also an alum of that program, and I was directed to her because of her mentorship. And I think that's also quite apt as we're thinking about the people that are on the journey that we're on as well. And I couldn't think of a better guide in terms of looking at that combination of excellent, engaged, and heartfelt patient care, and combining that with excellence in research as well, and guiding and realizing that this was a home that I could engage in bladder cancer as well as other genital urinary tumors. And as we look back under her mentorship and learning about how far the field had come already, and as our patient advocates and speakers had already talked about, that already this was the best time possible in some sense to have bladder cancer compared to all the advances that have been made recently. We still realize that there's still a long way to go, that there's still so many questions that are left to be answered, and we can be engaged in terms of doing those. And so that brought me to this field and to the projects I'm working on right now. And in particular, I've been working on rare bladder cancers. Again, as we talked about one of our guest speakers just now that had small cell bladder, squamous cell. These were areas where, as we're looking for the path, there are even fewer guideposts on what to do, given the limited participation in clinical trials. And also one of the things that I wanted to address through the research that I was doing. The projects that I'm engaged on, the preclinical models and clinical trials, are 3 projects that are all intertwined in that goal of both that research and clinical care. The first part is looking at in vitro and in vivo models using cell lines and available patient-derived xenografts for a number of rare bladder cancers, as well as a few other genital urinary cancers, particularly doing efficacy studies with antibody-drug conjugates like enfortumab vedotin, sacituzumab govitecan. Some things are already used, but trying to expand those for some of these rare bladder cancers, but also using this as a platform for which we could tackle and address whether or not they might be reasonable targets for other antibody-drug conjugates as well, and also to be able to generate new cell lines, new patient-derived xenografts, mainly from our patients at the NIH, that can be used for ongoing research both at the NIH as well as for researchers like many of the folks that are here right now. The second part of the project is a preclinical study looking at the expression of some of the targets of some of these main antibody-drug conjugates, like Nectin-4 and TROP2, from a range of these rare bladder cancers from multiple locations, from patients at the NIH, collaborators at Howard University, and in addition to one of our collaborating sites in Brazil, to get a wide scope of these tumors as well as ethnic and diversity that is present. Also, using these as a platform to have a better base of information about what might be reasonable targets to provide better ideas and guidance in the clinic. And the third part, and possibly the most exciting on my side, is the SMART clinical trial. This was an investigator-initiated trial that Dr. Apollo helped me guide from a concept all the way to the point of this week when we are just able to start enrolling and treating our very first patients after several years of work, to be able to have new options that we're actually testing in the clinic based on the information. Look forward to sharing this information as we go through. And so in summary, that's how I got to this point. And again, as we continue to think of ourselves guiding and being along the walk with the patients that we have, and working with the researchers here at this clinic and moving forward in the future. I look forward to meeting the patients, caregivers, families, and all those that are here that are engaged in that walk as well. And as we think about making sure that we have the right name, Beacon, there's nothing more apt about how we provide light for the future of how we provide guidance for everyone to improve the lives of folks impacted by bladder cancer. I look forward to meeting you in person, and please come and talk to me as much as you like. Thank you very much, and again, it's an honor to be here and to be here with the other fellows. Thank you.
Next, we have Dr. Manib Alam, who is a urologic oncology fellow at Memorial Sloan Kettering. His project is titled Neoantigen Quality, T-Cell Diversity, and Response to Intravesical BCG. His mentors are Dr. Eugene Pietsak, Dr. Hikmat Alamadi, Dr. Gopa Iyer, and Dr. David Solit, typical of Memorial to have so many mentors. Dr. Alam is an early career surgeon scientist and current SUO fellow. He completed his residency in urologic oncology at the University of Kansas. His research is focused on studying mechanisms of resistance and response to immune-based therapies in urothelial carcinoma through multimodal next-generation sequencing. He plans to pursue translational research as an academic urologic oncologist. In his personal statement, he says, by supporting the development of aspiring surgeon scientists like myself, the John Quale Travel Fellowship highlights Beacon's commitment to a bright future for bladder cancer research and advocacy.
Good evening, everyone. Obviously, a very great opportunity for me to be here, and I appreciate the opportunity to speak today by myself. So, without to belabor so much of what you've already talked about, I'm a urologic oncology fellow at Memorial Sloan Kettering, primarily focused on bladder cancer, and really have had an interest in bladder cancer for a long time. What got me into bladder cancer research was actually just looking for research opportunities in medical school, and I had a great opportunity to work with Dr. Taylor when I was a medical student. John Taylor's lab was at that time focused on mechanisms of resistance to neoadjuvant cisplatin-based chemotherapy, and so I had the opportunity to do some bench work, and that was really a formative time for me. I had one of my really first great mentors in urology, and that inspired me to kind of go forward into the field. I had a lot of great opportunities in residency. I always tell medical students and trainees that some of the best mentors across urology are really the ones that focus on bladder cancer, and I feel like I've really benefited from that. I'm now at Sloan Kettering doing my fellowship embedded within the lab of David Solit. You heard all the names of my great mentors. The project that I'll be kind of discussing tomorrow is really under the guidance of Eugene Pietsak with a focus on non-muscle invasive bladder cancer, specifically high-grade non-muscle invasive bladder cancer and mechanisms of response to BCG. When I first got to fellowship, I was very interested in looking at DNA damage repair gene alterations in non-muscle invasive tumors. These are very common, very important muscle invasive disease, particularly when it comes to response to cisplatin-based chemotherapy, but may also have an important role in non-muscle invasive disease. And some of our data showed that patients who had these tumors that had DNA damage repair gene alterations, specifically ERCC2, these patients had better response to BCG, and it appears that that's because of these the hypermutation that occurs in these tumors. Bladder cancer is already hypermutated, but these tumors have a really high tumor mutational burden. This led us to hypothesize that perhaps it's the neoantigens that are developed by these tumors that are immunogenic and lead to an improved response to BCG. And so based on that work, I've kind of moved forward. We've developed a collaboration with the Benjamin Greenbaum lab at Sloan Kettering. They've really kind of pioneered the concept of neoantigen quality. So these tumors, they have mutations, they develop neoantigens, but not all neoantigens are created equally. Some are more likely to be presented and received by the immune system, and so we really are interested in identifying those and understanding their characteristics. And so the first aim of this is really to look at the neoantigen landscape, to define neoantigen quality in tumors prior to the receipt of BCG. So we're doing that both in tumor tissue and also looking at the T cells in these specimens to understand T cell clonality, because that really is a reflection of the immune system's response to these neoantigens. And the second aim is also to look at neoantigens, but in the post-BCG setting, to understand what happens to these neoantigens In the setting of immunotherapy in the setting of systemic immunotherapy for metastatic disease, it's clear that there's a depletion of high-quality neoantigens following immunotherapy, and so we think this may happen after BCG. And that has an important role potentially for repeat therapy with BCG as well as using immunotherapies after BCG receipt. So looking at matched pre and post tumors to try to understand is there a depletion or selection for these high-quality neoantigens, both looking in the tumor tissue as well as in peripheral blood from before and after treatment to try to understand the expansion or contraction of different T-cell populations. So that's kind of the work that I'm working on now. It's been a really great honor to be invited to this meeting. I feel like it's been a long time coming for me. I've heard about it all the time from my mentors, and thank you for inviting me to speak today.
Next, we have Dr. Marie-Pierre Saint-Laurent. Her project title is, I think, my favorite. It's called Neoblast, Neoadjuvant Therapy for Bladder Cancer Followed by Active Surveillance versus Definitive Treatment. Her mentors are Dr. Peter Black and Martin Gleave. Dr. Saint-Laurent graduated from Laval University in Quebec City, Canada, where she obtained her doctorate of medicine and urology residency training. She is completing a fellowship in urologic oncology at the University of British Columbia and is beginning her third year of fellowship with a research focus on urothelial carcinoma. Her interest is in developing a career in translational and clinical trial research. In her personal statement, she says, "Receiving the Quale Travel Fellowship to attend the think tank is a tremendous honor that will not only help my research project in bladder cancer moving forward, but also provide me with invaluable networking opportunities. This will give me the opportunity to present my clinical trial concept on active surveillance for muscle-invasive bladder cancer patients post-neoadjuvant therapy. I look forward to receiving feedback and engaging with experts and patient advocates, and enriching my approach to patient-centered care and innovative clinical trial design."
Hello, everyone. My name is Marie-Pierre Saint-Laurent, but you can say Mary if it's easier to pronounce. I'm from Quebec. Did my medicine there, my urology residency, and now I'm in Vancouver. And I'm a little bit slow at doing things, so I decided to do a third year of fellowship just so I can do a bit more of research with a focus in bladder cancer. Really thank you for this award, and that fellowship grant is really nice to be here and have the opportunity to talk to you and just share briefly my interests. I'm trying to do multiple things. Hopefully I will succeed in some of them. I do have an interest in trying to understand a little bit more about bladder cancer as genomic and dipping into epigenomics. But here tonight I want to talk a bit more about the clinical research aspect I'm interested in. I'm happy to hear you like the name. It was really hard to pick a name for a clinical trial. Hopefully it will be also interesting to hear the conversation on Friday's session about bladder preservation in the meeting session. So I'm lucky to be in Vancouver. I'm under the mentorship of Dr. Peter Black. Maybe some of you know him, and really grateful to be there. And with him, we've worked to design this clinical trial. So what I'll be presenting tomorrow is maybe a little bit different, but it's really the protocol, the design concept of the clinical trial we're working on. And really the idea came out from seeing what's already been started in terms of clinical trials to offer de-escalation or perhaps more personalized treatment in some of those great responder and patients have muscle-invasive and receive neoadjuvant chemo. We know that 38% or 40% will have complete response at the time of cystectomy. The questions often when you give those results to the patient is like, well, did you really need it to remove my bladder? Like, could I have kept it? Of course, how can we know without having it under the microscope and the pathologist to tell us there was really no cancer left? So that is one of the good questions we all wonder. What are the tests that will really give us enough certainty that there is no cancer left in your bladder or elsewhere? So that's one of the first things in the trial we've designed, that we hope to incorporate the novel biomarkers we have. And then the second thing is, with all of those, blood, urine, MRI, will that be enough to say that we really do not need to do a treatment on the bladder? Will we still need to have trimodal radiation or cystectomy? So how can you really be sure? And you know, as the gold standard in the research is usually to do a randomized control trial. So can we do that? We know that sometimes during trial, when we do treatment observation versus surgery or radiation versus surgery, it's always been difficult to achieve. So we want to propose to do a pilot RCT, a pilot study, and see how feasible this is, and hopefully work with patients and get feedback and see how this is possible, and if so, learn from potential mistake or learn from success before we move for a larger randomized trial. So I welcome anyone's feedback during that, and please come see me and discuss any questions or idea you have. I'll be interested in hearing about it. Thank you.
Next, we have Dr. Catherine Marchetti. Her project is, she's a urologic oncology fellow at University of Pittsburgh. Her project title is The Use of User-Centered Remote Monitoring Program to Improve Patient-Reported Outcomes and Readmission Rates Following Radical Cystectomy, under their mentorship of Bruce Jacobs, Ben Davies, and Christian Stensland. Dr. Marchetti, as I said, is a SUO fellow at University of Pittsburgh. She earned her undergraduate degree in biochemistry and her medical degree as well at the University of Michigan. She also completed her urology residency at University of Michigan as well. Dr. Marchetti's research primarily focuses on the use of remote monitoring with wearable devices to identify and treat early signs of complications following surgical treatment for bladder cancer. This aligns with her overall goal of advancing the use of technology to improve patients' lives and interactions with the medical system as they navigate their cancer care. In her personal statement, she says, "As a urologic oncologist, I'm motivated by my experiences with patients to continually improve my clinical care. Connecting with clinicians, researchers, and patient advocates at the think tank will facilitate my research on the use of remote monitoring to improve the quality of patient care after cystectomy."
Hello, everyone. Just another thank you for the opportunity to come to the think tank, be able to learn from so many of you with diverse and unique experiences is really exciting to me. As was mentioned, my name is Katie Marchetti. I'm the oncology fellow at Pittsburgh and a Michigan lifer before that, go blue. But actually my hometown is San Diego, so what an exciting reason to be back. So first, I just want to thank my mentors that have helped me in my training so far, specifically for this research, Dr. Bruce Jacobs, Ben Davies, and Dr. Christian Stensland. But really, it's my clinical experiences, as was stated, that inspire my research, and so I need to take a moment to thank my many patient mentors who have intentionally and unintentionally guided my clinical and academic work. So why urothelial cancer? I feel like in training, often our bladder cancer patients would become familiar faces. They would be the people you'd see in clinic multiple times for their BCG installations or on rounds when they needed a little extra TLC after cystectomy. And it's these familiar faces that really trained me to be able to take care of patients with bladder cancer. But despite this familiarity, many of them would leave me confused. How would I know what one patient does well, another doesn't? How could I have known their path report would have showed what it did? And it's this uncertainty that really drives my interest in bladder cancer and my specific project that I'm going to discuss today. So reflecting on the stories of these familiar faces after cystectomy, those that were readmitted, many of what they told you sounded the same. They would leave the hospital and they felt great. And then they'd come back in the ER a couple days later. They would perform your instructions and follow your advice to a T. What happened during that time between when they left the hospital and when they were readmitted? And so that's what I would like to use remote monitoring to discover, is what happens during that time. I want to use remote monitoring to listen to people in a time when they're particularly vulnerable in their recovery after cystectomy. And so I'm going to work to use user-centered design to improve the way that we monitor patients around the time of surgery. We know remote monitoring is feasible following cystectomy. It has yet to be shown to be efficient or effective at reducing patients' symptom burden and improving the rates of readmission. We have very powerful consumer-grade wearable devices that are accessible and easy ways to measure physiologic biomarkers in the outpatient setting. And clinical trials that have used remote monitoring with wearable devices have been successful in other areas of medicine, not in our post-cystectomy population. And this is probably because the post-cystectomy period represents a very unique time. Patients have distinct characteristics and challenges that render them uniquely vulnerable, and thus in need of a very intentionally designed remote monitoring intervention. And so as considering this, I aim to use methods of user-centered design to develop a remote monitoring program that keeps our patients' perspective at the forefront. I aim to incorporate wearable monitors into the way that we use remote monitoring today to create a program that serves our patients and augments the way that we're able to quantify their progress after they leave the hospital and bring us closer to be able to use remote monitoring to produce meaningful clinical outcomes in that period. In the last year, I have built a remote monitoring program with the help of the Mobile Sensing and Health Institute at the University of Pittsburgh under the mentorship of Dr. Chris Lowe. Really proud of this program. It includes patient-reported outcomes assessment via a smartphone application, as well as remote biomarker assessment with a wearable device to provide a two-dimensional kind of picture of our patients after they leave the hospital. And this program uses real-time provider feedback. It alerts providers to changes in these remote monitors in real time and is able to kind of start to incorporate some immediate feedback to those changes in vital signs and severe symptoms patients are having. So first we're going to be, as I mentioned, obtaining qualitative feedback from people, integrating that as the backbone of our process of user-centered design into modifying our remote monitoring program. And then we're going to utilize that program, that patient-centered program, in a pilot trial evaluating the feasibility of integrating these wearable devices and the real-time provider feedback into practice. So ultimately, I hope to access the barriers of scaling this program to multiple sites with implementation science, ensuring that our program fits the diverse needs of bladder cancer patients and providers throughout the country. So I invite you to come say hello. Please let me talk more about user-centered design, the use of wearable devices, our kind of the remote monitoring program we have set up. And I would love to get the advice from the think tank about how to ensure the success of my research team as I embark on a really exciting year. Thanks for letting me kind of kick it off with participating here at the meeting. Thank you all so much.
Next, we have Dr. Eva Michaud. She is a postdoctoral fellow under the mentorship of Dr. Wes Kassouf. Her project title is Gut Microbial Drivers of Response to Bladder-Sparing Therapy in Human Muscle-Invasive Bladder Cancer Enhances Immunotherapy Efficacy and Limits Immunosuppression Following Fecal Microbiome Transplantation in Vivo. She's a dedicated researcher specializing in immunology and its implications in cancer therapy. Dr. Michaud pursued postdoctoral research at McGill University, focusing on immune and microbial determinants of response to radiation therapy in muscle-invasive bladder cancer. Her work explores the interplay between the gut microbiome composition and the immune response in bladder cancer, shedding light on potential avenues for improving treatment outcomes. In her personal statement, she says, attending the Beacon think tank this summer will not only enhance my understanding of the latest advances, but also enable me to collaborate with leading experts in the field. This fellowship empowers me to translate cutting-edge research into tangible benefits for bladder cancer patients, bringing us closer to more effective and personalized treatments. I'm deeply grateful for this recognition and excited to contribute to the collective effort of improving outcomes for those affected by bladder cancer.
Hi, everyone. I want to thank you again for the honor to be here. My name is Eva. Any of my student would tell you it's a grave mistake to give me a platform and a mic, because I now have the power to talk to you, to your ears, for as long as I want until I'm forcibly removed about what I like to work on. But funnily enough, I was that overachieving undergraduate future scientist who kept saying, cancer, it's getting all the funding. I don't want to do it. I know it all. I know it best. I'm 19. More than a decade later, I'm in this room and couldn't be prouder. So I always wanted to study immunology, actually, and so my path was very immunology and health science oriented. And I did my graduate degree in fundamental immunology, and in doing so met the person who would mentor me in my doctoral studies. who was a lead scientist for mucosal immunology. And in studying the particular aspects of mucosal barriers, I realized that, we understand immunology in terms of like vaccines, et cetera, but what's happening at mucosal barriers is not only you have an organ that has a function, say the gut, it helps you digest, and there are key relationships happening in this organ that are defining for your organism in terms of immunity and in terms of interaction. And so the microbiome that you have, the nice bacteria that sit on your mucosal lining, they're not just sitting there. They're really providing education for your immune system. And so that's what I learned in doing a PhD there, and I realized we have to have a vision as scientists and more like systems biology oriented. We're too used to looking at a cell and this organelle and this protein. But I wanted to take a step back and be like, okay, what happens in other mucosal surfaces? And it turns out we know a lot of the lung, we know a lot of the gut, and I was looking at other niches, and I realized the bladder, it's the same. It's a mucosal organ. It has the microbiome. We know that now. We don't really know what happens there. And so I was lucky enough to meet Dr. Kassouf for my postdoctoral degree, postdoctoral years. And we started working on bladder cancer. It's particularly interesting because you have these relationships that are not described in the bladder, but now in bladder cancer, you also have a tumor there which is probably going to affect these responses. So all in all, what I did for this lab was I took the microbiome project, and we first looked at gut microbiome, because we know gut microbiome, even from a distance, it can condition a lot of things. And what we set up is we were like, okay, we want the patients to keep their bladders. We also know that sometimes that, bladder-sparing therapy, radiation doesn't always work. What if we combine it to immunotherapy, but that also doesn't always work? So what if there's something, conditioning those not working responses? And so we took patients that had responded and not responded. We took their microbiome and gave them to germ-free mice. And lo and behold, the mice didn't respond very well if they had received non-responsive microbiome. And that's the only thing I changed there, because I'm really a psychopath scientist. So I was very controlling in terms of variable. I was the only one touching these mice. There's only another technician that was like, really what we only changed is that microbiome. And it came from patient. And if we gave them responsive microbiome, of course, they were responding very well to radiation and radiation combined to immunotherapy. And when I dug a little bit deeper, we find that in these mice that respond very well, what is actually happening is they have this population of T cells that is switching to effector function. Now, that same population is also in the non-responsive mice, but they're just not switching. And so I've never seen that data. I've never seen it in bladder cancer. I've never seen it in other cancer yet. But what we have is we have responsive microbiome that are enabling a switching in T cells from a distance. And I found that particularly interesting. And so as I'm transitioning to more lead investigator roles now, I want to focus a little bit more on that in the bladder. What's the contribution of the bladder microbiome to these relationships, but also what's the contribution of these relationships to sex differences, because we've seen that there's a gender disparity, a sex disparity, should I say, between males and females, and I would like to observe that through the prism of host-microbe relationship. Okay, you won't have to forcibly remove me. Thank you for listening to me. I really look forward to exchanging on those topics and any other. Because we had the patients advocacy panel right after, I'd also like to highlight that I'm just a research person here. I'm not a clinician, and what I found in my, career leading up to being here is we are also not very well sensitized to what it means to have the disease that we're studying. I can tell you anything you want about the cells of these tumors, but really I don't know much about the patient aspect of it, but I also knew very little about the clinical aspect of it. A lot of the terms that have been used just now, I'm like, oh, I thought I knew it all, but apparently there's some, so I think there's also a dialogue that's not always happening between the scientists, the clinician, and the patient, and I'm very glad to see some shadowing programs pop up in many universities so that researchers like me can follow clinicians around and see what's actually happening in practice. So thank you so much again for this opportunity.
Next, we have Dr. Shilpi Chaudhry. Her project is titled Deciphering the Bladder Cancer Immune Microenvironment Dynamics in Genetically Engineered Mouse Models with Epigenetic Dysregulation. Her mentor is Quale. Dr. Chaudhry is a postdoctoral research scientist at Columbia University Medical Center. Her research is centered on elucidating the implications of epigenetic dysregulation and their impact on tumor immune microenvironment using in vivo genetically engineered mouse models. Dr. Chaudhry earned her PhD in medical oncology from the All India Institute of Medical Sciences in India. Her goal is to identify the targeted therapies for bladder cancer patients with epigenetic gene mutations for potentially improving the treatment outcomes in patient care. In her personal statement, she says, "Receiving this travel award is a significant milestone for my career. It provides an invaluable opportunity to share my work with leading experts, fostering collaboration and innovation. This recognition will advance my professional development, but also bring me closer to breakthroughs that can improve the lives of bladder cancer patients."
Okay. Hello, everyone. I'm Shilpi, second-year postdoc from Columbia University. I'm really honored to be here today, and I'm really thankful to Beacon for giving me this opportunity to attend this meeting. So, starting my journey, I did my PhD from India in medical oncology, where I was learning about the acute myeloid leukemias, and we evaluated the gene signatures which are responsible for predicting the adverse outcome of the patients. So at that time, I realized the importance of clinical research and the translational research, where we can, how it would be beneficial extremely beneficial basically for the patients directly implicated to the clinical trials, and helpful for the patient care. So after completing my PhD, I came to Columbia University. I joined Quale lab, who's very renowned in the bladder cancer field. So what we do in our lab, actually we know the bladder cancer is a very interesting disease, having a lot of mutation burdens, and especially the epigenetic dysregulations is more common in more than 30% of the patients, but there is no targeted therapies for particular patients having epigenetic mutations. So in our lab, we have developed a series of genetically engineered mouse models which mimic the epigenetic dysregulated patients, and they develop muscle invasive bladder cancer. So these models, epigenetic gems, are very useful for identifying the molecular mechanisms of how bladder cancer initiates and progresses to non-muscle invasive to the muscle invasive disease, and later on to the metastasis. So our goal is to identify the targeted therapies for the patients having epigenetic dysregulations, and I am here to collaborate and get all the valuable feedback from the experts of the bladder cancer field. Thank you.
Wonderful. Our final fellow is Dr. Charles John. His project is titled Care Access, Quality, and Outcomes Among Patients with Advanced Urothelial Cancer, a comparison between Medicare Advantage and traditional Medicare. His mentors are Dr. Lau and Gerber at UT Southwestern. Dr. John is an assistant professor at UT Southwestern Medical Center as a medical oncologist specializing in the treatment of bladder and other genitourinary cancers. He holds his MD from Soochow University and MPH from Yale, and completed his residency at Mount Sinai and his fellowship in medical oncology at Roswell Park. Dr. John's research focuses on health services and policy, particularly examining the impact of health insurance and social determinants on cancer care access and outcomes. He says in his personal statement, "Receiving the travel fellowship is a tremendous honor that will propel my research on healthcare access for bladder cancer patients. This award enables me to focus on breaking down barriers to high-quality bladder cancer care, making a real difference for those in need. Thank you, Beacon, for supporting this vital work."
Thank you, Beacon, for inviting me and bringing me all the way from Texas heat. I'm really appreciative. Dallas is just so hot in the summer. But But anyway, it's great to be here. I'm Charles Jiang. I'm a medical oncologist. I just finished my first year faculty job. I'm very happy and with my research and my patients. The reason, and I do research about care access. I'm the access guy. I'm not a scientist. What I really care is, despite all the science advancement and clinical advancement, we still have a quite proportion of patients did not get the standard of care. They're getting suboptimal care somewhere due to all different reasons. And these patients, they may not be aware of a lot of existing resources. They may not advocating for themselves because they never understand to or they don't know how. And that matters. Why bladder cancer? I would say bladder cancer is very personal to me. My grandfather had bladder cancer. And the ironic fact is he was a surgeon. Back in the days, people were not specializing that much. He actually did quite amount of cystectomy himself. When facing a bladder cancer muscle invasive disease at that time, 15 years ago, he just told the whole family the things. I don't want chemotherapy. I know it's terrible. I saw patients suffer from that. I just don't want it. Thinking backwards now, at myself being a medical oncologist, I realized that may not be the best option for his outcome. But he's from the older generations. He makes his decisions. We respect that. But it made me realize, if you look at bladder cancer, it's actually one of the cancers that require the most collaborations between the specialists. We really need the whole teams, medical oncologists, urologists, radiation oncologists, nurse, and patients. Everyone have to work together to achieve the best outcome possible. And we just know many times it may not be possible for all different kind of barriers. And why specific about Medicare Advantage? I just got into this. I hit the gym other days, and I saw this TV commercial, and I probably just ask around here. Raise up your hands if you ever seen a TV commercial saying, oh, call this number so you can get this free Medicare insurance, and they give you the free gift card and all the things like that. Have you ever wondered what happens if you call these guys? We don't know, right? And I'll tell you, as an access guy, as someone who does insurance research, I don't know either. But guess how many people are insured with Medicare Advantage? According to most recent data, in 2023, 50% of the Medicare beneficiaries now are under coverage of the Medicare Advantage, while we don't know anything about these plans and how that impacts cancer care. And if you actually go to the insurance website, I'm just not one of the insurance companies, and you say it's like, are these free insurance true? What's the catch? And they'll tell you, they'll lower the out-of-pocket cost, they'll lower the this and that cost. You're going to only suffer a little bit. You're going to only see certain doctors in the network. And what that means, right? Nobody had cancer before they diagnosed with cancer. And you probably will never realize that. You need to find an in-network doctor, and you don't even know how many oncologists are in that network. Actually, CMS, the people who are paying the biggest payer in this country, the big payer for Medicare, actually they have certain regulations. They say, oh, if you want to put up with this Medicare Advantage plan, this plan using the taxpayer money, but operated by the private companies, and they have to meet certain regulations. This company have to show to the Medicare, to CMS, that they have certain amount of the primary care doctor in that network. They have certain amount of surgeons in that network. Unfortunately, oncologist is not one of the specialties. And it probably means more to a bladder cancer patient because first, bladder cancer patients tend to be older. Second, we need a good team-based work to achieve the best possible outcome. So here's my research. What I do is I use a real-world database. Flatiron Health basically is a database getting the data from 800 more different care sites, and they're pumping the data into this database. They are all real patients with bladder cancer, and I'm just going to look how these patients are doing and what Medicare Advantage means to them. Are they getting the care timely? Are they getting the care they're supposed to get? And are they having the good outcome they are supposed to get? And are they getting all the next generation sequencing they're supposed to get? It's an ongoing process, an ongoing project, and I have some result presented. And so, welcome to say hi to me if you have any experience regarding any insurance, no matter if you're a doctor, patients, or industrial partner. I'm very happy to hear that. And please say hi to me. Thank you for bringing me here.
All right, thanks everybody. Hopefully you've realized that that the bladder cancer research field across the spectrum is alive and well, and I really hope that you go up to our fellows and to each other who you don't know. That's the whole idea of the think tank in general, but especially to our fellows, and engage them in their work and in their lives. So now we're going to have. Are we going to have a break, or do you want to? Okay, perfect.
Thank you again so much, Max, for your leadership of that program, for moderating the session, and thank you to all of our fellows for sharing your research with us. We are excited to see your posters tomorrow. We welcome you to the Beacon family and hope to see you at many more think tanks.